Hello and good morning. My name is Drake Roush, and today I'll be sharing with you the polymerase chain reaction using lab DNA. Special thanks to my partners Harry Zhang and Margot Starr, as well as our teacher, Dr. Shingleton. The purpose of this lab is to practice the PCR techniques in a system that'll work reliably before we try it on our own DNA. This process is useful because PCR can be sensitive to imperfect procedures and DNA can be easily contaminated. PCR is a method widely used in molecular biology to make many copies of specific DNA segments. Using PCR, a single copy of a DNA sequence is exponentially amplified to generate thousands to millions of more copies of that particular DNA segment. The reason for this lab is that the class will have our first opportunity to magnify DNA using the PCR. The importance of knowing and mastering this method is because it allows us to amplify a targeted region of a single molecule of DNA into as many exact copies as we want. PCR mimics DNA replication that takes place inside the cell. However, PCR takes place in vitro. It takes place outside of a living thing. In this lab, we will increase a region of DNA from the bacteriophage known as lambda, a common virus known for attacking bacterial cells. Before you do anything, make sure to put on those gloves to protect reaction from oils on your hands and other contaminations that could affect the results of the lab. To start off the lab, you will cast the agarose gel in the buffer TBE 1%. This is a different gel than the last lab when we used a TAE gel. As you can see, we are using electrical tape to act as the border to surround the gel. We use TBE because it's a strong inhibitor for many enzymes involved in the process of duplication. After the gel is complete, it's lab time. We just want to note that in our class lab, there was one control group for every 2 groups of people. The control group will contain the same contents as the PCR reactions will. However, it will not be cycled through the thermocycler. Therefore, the temperature wouldn't change. Before you begin the procedure, be sure to label your name and make sure to label the control tube in a different way. Once you are done setting up, place TAQ, polymerase, ready to go, bead, nucleotides, and salt that are necessary for the reaction into the PCR tube. We use Taq polymerase because it's a synthetic enzyme that produces DNA strands at a faster rate than natural polymerases. Once this lab is accomplished, place the tubes into the cooler containing ice. It is essential to keep your tubes cold since low temperatures protect the DNA by slowing down the activity of enzymes that could possibly break it apart. Note, the cooler will act as a resting place for the tubes. After this, firmly tap the tube onto a hard surface to make sure the solution goes to the bottom of the tube with the PCR bead. It is important for the bead to completely sink in the solution. Then, using a micropipette to the 20 range, add 20 microliters of the primer mixture, which is a short single strand of DNA that serves as a starting point for DNA synthesis. Then, add 10 microliters of lambda DNA into the tube. Make sure when you're doing this that the DNA lambda is directly hitting the solution. Once all of the solutions are into the tube, please remember to firmly hold and flick the tube to mix the solution as perfectly as possible. After all of this, we gave our tubes to Dr. Shingleton, who would place the tubes on ice with the rest of the class's tubes. She would place them inside the thermocycler, however the control would be placed on a rack without the ice. And there you have it. Day 1 is complete. Welcome back, welcome back. It's time for day 2. You know what day it is, the day to run your gel. To start this lab off, you want to add 4 microliters of UV loading stain to your PCR tube. You also want this in your standard. Traditionally, for the DNA to be seen on a gel, we would have to use a technology called staining, which takes a lot of time to accomplish. UV stain was best for this lab, as it serves both as a visual guide for the electrophoresis and will fluoresce under the UV light to show the bands after the electrophoresis is complete. And do the same for the control tube. And of course, close and flick the tube right now. Then load approximately 15 microliters of PCR loading dye mixture, which allows the scientist to see the speed of dye migration in a running gel. And make sure to use a different lane for each sample. After that, you want to load approximately 12 microliters of the control we shared with the other group members. And then load 15 microliters of the size standard that was provided by Dr. Shingleton. A size standard would later act as a reference for us to measure the size of all the other samples. Poured all of the solution, it's time to run your gel at 120 volts, which is the optimum voltage for our gel, using electrophoresis chamber. Once the loading dye has moved 45 millimeters away from the wells, your gel is complete and it's time to take photos. Sadly, our gel didn't get the results we were hoping for because we did not have the right volume, or the standard wasn't stain dark enough because it either needed more DNA or more UV stain. Because of our mistakes in the lab, we decided to borrow results and pictures from other groups to help us with the conclusion and discussions for our lab. If you want to see our full discussion and conclusion of the lab, check out our Weebly. Thanks for watching.